All right. So thank you everyone for being here, for joining us for this first part of our interactive virtual mini workshop on doing viral infection using CompuCell3D. I'm TJ Sego. I'm a postdoc at the Biocomplexity Institute in Indiana University, which is directed by Professor James Glazier, who's joining us today, as well as Josh and Giuliano, who are doctoral students in our lab. And then lastly, Dr. Gilberto Thomas, who will be joining us from Brazil. So today we're going to start with some of the background on some of the relevant biology that we're particularly interested in concerning modeling viral infection and SARS-CoV-2. After that, we'll do a sort of flyby of the model that we've developed that our simulation framework implements, and kind of blast through some of the key results from the paper that we've recently submitted using that. After that, we'll take a break. We'll do a Q&A session, and then take a quick break for anyone who needs to rehydrate or caffeinate or whatnot. And then after that, then we'll come back and we'll really dive into playing with the model. Today we're going to stay out of the code for the most part, and instead we're going to mess with some model parameters and observe what we see in the infection dynamics that emerge from the simulations. So the way that we've set this up, just to keep everything moving smoothly, so we've disabled screen sharing and microphones for participants. So we would ask that for any questions or comments or concerns to enter those into the chat in Zoom. We've set up some breakout rooms, and James, Josh, Giuliano, and Gilberto are happy to help. So if there are any issues that should arise, particularly with installing CompuCell3D or launching it in NanoHub or anything to that effect, just let us know in the chat. We'll set up a breakout room and can work one-on-one with someone to resolve whatever issues you might have. So, as excited as we are to be here today for an event like this, it's sort of bittersweet that we have to start by talking about such a dreary fact, which is that we do live in quite sobering times. I'm calling from my own personal jail cell of quarantine at home, as I'm sure most people are as well. And this is sort of the nature that we have right now. This is our current reality. And we as modelers come into this wanting to know what can we do to help. How can we contribute to understanding what this virus does and how to mitigate its effects. And it's particularly tricky and a difficult bug to deal with for a lot of reasons, particularly that patients tend to be infectious before they're symptomatic, that the outcomes between patients who are infected can vary a lot, not only from person to person, but also by location, and also concerning the causes of death. So there have been many reported causes of death due to complications associated with COVID-19, and those range from cardiovascular issues to respiratory issues and so on and so forth. And then there's that most of us are coming into this very early with respect to when all of these events occurred, and that the data to really even try to predict what this thing is doing is very sparse. It grows by the day. It's very difficult to keep up with, but by and large, we sort of have to work with what we know based off of other viruses like influenza that are better understood, with the hope that over time, as the body of data collects, that we can fill in the pieces as the data arrives. Now, there are a lot of questions that motivate modeling COVID-19 and SARS-CoV-2 infection. A lot of questions that we as modelers would like to answer, particularly concerning understanding both how this thing works and how we can deal with it. There is the significant variation between people who get either mildly or severely sick compared to, I think, what are called the silent carriers who carry the virus but show no symptoms. We'd like to understand why this virus behaves differently with respect to other viruses that we understand much better, again, like influenza. We don't really understand a lot of the mechanisms for why there seems to be such a delayed adverse response in individuals and what causes complications like pneumonia, cytokine storm. We also seem to not really understand much about age-related effects and how that contributes to determining patient outcomes, as well as the spatial differences between different areas of, or different regions of tissue, say where infection either begins or where it progresses to. Now, for more of the biomedical applications, what we'd really like to know is if we could use modeling and how we could use modeling to, say, better inform for triage of patients, or how we could help in drug development and drug discovery by assisting in optimization of antiviral and anti-inflammatory therapies. And what we'll go into today, and what we tend to be particularly interested in with doing this sort of modeling that we do, is whether or not we can predict system level effects due to molecular level perturbations. So if we change a particular lower level, sort of subcellular level mechanism, what are the observed emergent effects all the way up to the systems level? So to do this, we need to go through some of the relevant biological components to something like a SARS-CoV-2 infection and COVID-19. And there are, unfortunately, a lot of mechanisms, a lot of biological components that tend to be involved over lots of scales and locations throughout the body. But most immediately available, we have the epithelial target tissue. So an epithelium that is subject to exposure of a virus, viral internalization, viral replication, and so on and so forth. And then, of course, the immune cells in the epithelial tissue that are responsible for clearing the virus and eliminating infected cells. And that introduces a lot of complications and complexity concerning how they're recruited to the local tissue and site of infection, as well as what specifically each particular cell type does. There's a lot of complexity concerning the immune signals responsible coming from a local site of infection and resolving to recruitment of immune cells to the site. Then there's the consideration of damage, cell death due to replication, cytotoxic killing, so on and so forth, and a positive outcome for a patient, local tissue recovery. Then there's the systemic immune system and the immune response more at the global level that has to do with the complexity of the immune signaling from a local site of infection into more of a global scale, the response that tends to resonate throughout the body. And this involves lots of complexity concerning proliferation and maturation of immune cells. Now, getting to and from a local site of infection and a systemic response involves then lots of complexity concerning whole body transport with the cardiovascular and the lymphatic systems. There's also the consideration of something like gas transport in air with initial exposure to a viral agent. Now, we're also somewhat interested in concerning some of the complications that can arise that lead to fatal outcomes, something like secondary target organs, and where complications arise, say for death due to cardiovascular failure and whatnot. And then lastly, there's the general complexity concerning the various stages of the immune response with respect to the early innate immune response versus the later adaptive immune response that tends to be more specialized to a particular pathogen. Now, concerning the immune response, and the various components that go along with it, the immune system behaves very particular, not very particular, it behaves particularly to a particular pathogen. And so what we try to do for now is to describe the general behaviors. And so a figure like this on the right is something that we've constructed as an aggregate of a number of diagrams for a number of different viral agents to sort of convey the sequence of events from the moment of infection all the way through the first couple of weeks. So the early innate response, which occurs typically in the first 5 or so days, tends to begin with immune cell scavenging and phagocytosis by macrophages. And this can occur pretty much immediately and lasts through the first couple of days. There's interferon response and antiviral signaling from infected cells, as well as systemic cytokine signaling that begins around the first 2 days. And that's sort of amplified and relayed by local immune cells to gear up the immune system into the adaptive response. There's then the recruitment of natural killer cells that tends to start pretty fast and peaks somewhere between 3 and 5 days. And then while this is going on, then there's the migration of dendritic cells to do antigen presentation in the lymph node, which occurs around 4 to 6 days. This results then in the maturation and the return to local site of infection of CD8 plus T cells that do cytotoxic killing or specialized to a particular pathogen. And then there's the later B cell proliferation and then the production of antibodies resulting in an adaptive immune response and inevitably, in most cases, a long-term immunity. So when we look at the virus and what the virus actually does, it has its own sorts of complexities. So in something like SARS-CoV-2 or also in influenza, if a virus that's introduced to an epithelium or a particular cell population, and what we'll find is that there's an eclipse phase, usually somewhere in the first 6 hours or so after exposure, where there really isn't any actual release of the virus from the cell population that's been exposed. But then after this time, you can see in both of these diagrams, there's then this exponential growth of viral production and release into the surroundings, after which then there's this sort of saturation that you see here, which is a result of the infection being mitigated by the immune system. And then again, in a positive outcome in a patient, then you see this rapid decay that occurs after this saturation. And this is the adaptive immune response, clearing the virus from the population. And so in these sorts of outcomes, there's then the corresponding effect in the epithelial tissue itself, which is damage, and in the positive outcome, tissue recovery. So cells can die from lots of different mechanisms associated with viral infection, both due to the presence and activities of the virus during viral replication, as well as antiviral signaling, like from interferons, to induce apoptosis in neighboring cells, and then cytotoxic killing by immune cells, both with surface receptor interactions, and then bystander effect, and then the secretion of reactive oxygen species. And then during recovery, usually somewhere around 10 days, at least in the case of influenza, then proliferation begins due to stem-like cells in the neighboring tissue. And so from this, we can make this fairly short list of the biological components that we're particularly interested in, and mapping those onto model objects, mathematical objects, and so on and so forth. And these are then at the levels that we're interested in: epithelial target cells, viral replication inside target cells, immune cells that are doing actual, say, cytotoxic killing and scavenging, and then a systemic immune response that's associated with more systems level faraway lymph nodes. So when we look at a lung epithelia, there are some very specific biological concepts that are relevant to what we'd like to model. First, that an epithelium tends to be approximately quasi-2D if we look closely enough, even though they do tend to come in tubular architectures and whatnot. That they vary in the phenotypes of cells that make up the tissue, if the geometry can vary or does vary over a particular spatial scale, and that the cells vary in their ability to be infected. So there's selective susceptibility and heterogeneity among the cell distribution. Now in our base model, we take that and we apply the simplification that for a particular tissue patch that we're going to simulate, that we're going to begin with a quasi-2D sheet of epithelial cells that are homogeneous, and they have a uniform susceptibility, and we'll simulate what goes on in this simplified sort of local tissue patch. Now for viral replication, there is a lot of complexity that goes on inside of a cell. And that complexity is specific to a particular virus. But what we do know is that there are the mechanisms of a cell undergoing internalization of a virus from its extracellular environment. We know that after that occurs, that there is then genomic replication, in the case of SARS-CoV-2, RNA replication. Particularly, this is exponential replication within a cell. And then the virus, in turn, is released from the cell, in the case of SARS-CoV-2 by exocytosis. And then, considering all that, then there are also all of the observations that we know at the higher level of the eclipse phase. And what we'd like to see is the eclipse phase, exponential growth, and then saturation in a model that we might generate to describe it. So in our case, the model that we've done is we have a system of 4 ordinary differential equations that do include an eclipse phase, as shown here by this dotted line. These are dictated by model parameters. And before this, there is no viral release due to the internal workings of replication. But then after that, you can see that there is this exponential growth and saturation. And during this time, then there's also a corresponding release of the virus into extracellular environment. The immune response has a lot of cell types and a lot of signals, and there's a lot of complexity, and in a lot of cases is still not well understood. But there are quite a lot of things that we do know occur, like scavenging and phagocytosis by macrophages. There's viral resistance that's induced by interferon signaling. There are killing mechanisms that immune cells do, like contact cytotoxicity, bystander effect, and then the secretion of reactive oxygen species. This involves a lot of complex signaling activities over time and over various scales. This involves a lot of antigen presentation and transport through the lymph nodes at the systems level. Those signals are relayed and amplified with cytokine signaling. There's a lot of complexity with the pattern of entry of cells and infiltration by immune cells into the tissue at the site of infection. In the case of both SARS-CoV-1 and SARS-CoV-2, there's a lot of depletion of resident T cells and macrophages, which last I heard is still not well understood. We know that both SARS viruses can infect both T cells and macrophages. There are a lot of non-specific antibodies that may play a role in early innate response. There's all of the complexity of both the innate and adaptive phases of the immune response and how one ushers in the other, as well as all of the complexity involved with the pro and anti-inflammatory signals that make all of this happen. So in our case, we elected to start very simply, which is to say that we have an immune cell type and one cytokine signal. And with those two generic representations, we've assigned, say, for the immune cells, the ability to do direct cytotoxic killing on contact with infected cells. We include a representation of killing by the bystander effect, as well as a secretion of a reactive oxygen species. Our generic cytokine field does something like activation of immune cells to do specific functions. And then for our quasi-2D simulation, we have our two-dimensional epithelial sheet. And then on top of that sheet, or below if you prefer, then we say that then when immune cells enter into the domain, that those immune cells rest on top of the epithelial sheet. Now for the systemic immune response, we know that local signaling recruits circulating immune cells as they travel by a particular location in the body. We know that these signals are very complex, and it's both a mixture of pro and anti-inflammatory signaling. And those signals end up undergoing long-range signaling that's upregulated by, to do immune cell production. Now, some of this also involves the migration of antigen-presenting cells, like dendritic cells, from the site of infection to a lymph node that's far away in the body. This results then in the production of CD8 plus T cells that are specific to the antigen. It also results in the production of B cells, after which then there's the production of antibodies that takes time over the scale of weeks. And then from that, there's the long-term immunity. Now we represent this with a single ordinary differential equation that describes the net pro or anti-inflammatory response or state of the systemic immune system. Now this represents the long-range signaling where the lymph node that's represented by this compartmental model supplies immune cells to the spatial domain of our simulation where our infected epithelial cells are. And this occurs when the immune system is in the pro-inflammatory state, whereas when it's in the anti-inflammatory state, then it removes immune cells from the spatial domain. And then we do this making sure to model something that represents a notion of variable delay. Now for tissue damage and recovery, we know that there are lots of killing mechanisms. They're both intrinsic and extrinsic pathways that lead to cell death from viral production, as well as all of the immune cell killing mechanisms that were discussed before, and then resulting recovery of the epithelium after an injury due to proliferation by resident stem cells. Now, in our model, we do have a representation of cells dying, which you can see here with this little black patch. And we assign the event of cell death as being due to the various cell death mechanisms that we represent. And then for now, for the given range of time that we've been simulating, we've not then introduced any notion of recovery into the simulations. And so when we consider these mechanisms, what we're interested in between local infection at an initial site of infection and what goes on in each individual cell and the recruitment of immune cells to the simulation due to signaling, you see that this spans three very distinct scales. So we have a sort of global systemic scale where the total cellular population exhibits signaling upwards in scale to the systemic level, in which case there's a corresponding response back to the cellular domain whereas when we look into each individual cell, we have a running model of the current state of infection inside of the particular cell. So we do all of this in CompuCell3D, which we here at Indiana University develop. So CompuCell is an open source simulation virtual tissue environment. So we do lots of training, both in using the methods that we provide in the simulation environment, as well as developing the simulations or the simulation environment. One of our main goals is to allow researchers, both in clinical and industrial applications, to develop models on their own without necessarily requiring any sort of expertise in computational modeling or computer programming. So we try to make this as accessible as possible for doing model specification, implementation, and interrogation. So for more info, this is our website, it's CompuCell.org. See in the chat that there's lots of directions on and discussion on doing installation, so you can find directories there. We'll get into that a little more later. So we do this because computer simulations that can explain how mechanisms interact and how they lead to specific outcomes. These focus on problems where the multiscale interactions are critical and where something like a molecular signature is insufficient to explain a more systemic response. So we look at specific mechanisms and how variations in their behaviors result in higher level responses. But we do this to understand disease mechanisms better, to identify critical pathways. We do this to try to identify drug targets for conditions that are currently untreatable. Particularly interested in doing personalized medicine and personalized improvements to existing therapies, as well as to identify toxicity mechanisms. So this is just a very brief list of some papers that have been done by topic. So see, like there's polycystic kidney disease and lots of work that's been done there. We have ongoing work in diabetic retinopathy, as well as liver toxicity. Now, one thing that is missing from this list is viral infection. And this is the preprint that we have developed to document the work that we've done so far, to convey and communicate what's in the simulation framework in its current state that we'll go through today. We've submitted this to PostCompBio, and that's all in the pipeline concerning that. But what we would like to see is that the framework that's presented here and described in this paper, that this is sort of the launching point for this new field of study using CompuCell3D based modeling and simulation. So I'd like to go through this as quickly as we possibly can without diving too much into the mathematics and all of that, but instead to more so go through and get a sort of higher level understanding of what's represented and what we can play with in simulation currently concerning parameter variations and simulating scenarios and whatnot. So our strategy with developing this model was to first start with initial infection in a small patch of susceptible tissue. So we assumed a scenario of primary infection where there's no previous exposure to the pathogen that's being simulated. And so therefore no specific antibodies, no memory T cells or B cells. And then we assumed acute infection. So we're simulating a relatively short amount of time and looking specifically for outcomes where either the immune system clears the virus or widespread infection occurs throughout the entire tissue, or something in between. So this model consists particularly of two cell classes, those being a generic epithelial cell and a generic immune cell. Now the epithelial cells can be infected, and the immune cells fight the infection. We also consider three diffusive fields that we simulate along with the spatial distribution of these two classes of cells. The first being a representation of extracellular virus transport in the extracellular space. There's then also our generic cytokine field that we use to map the effects of both local and global signaling by infected cells and immune cells. And then secretion of an oxidative agent, which is something like a reactive oxygen species that immune cells can produce to kill epithelial cells when activated. And then again, we have a compartmental model that we use to represent the systemic immune system, describing either the pro or anti-inflammatory state of the immune response. Again, this is a compartmental model using an ordinary differential equation, and we use this to regulate the local immune cell population in our simulations based on the signaling that's done within the simulations. So our epithelial cell types, we have, again, one class of epithelial cell, but we describe it as being of one of four types. Now, these are not biological types in the sense of it being an epithelial cell or a particular epithelial cell, but instead we use it as an identifier for the various stages of infection in a particular epithelial cell. So if an epithelial cell has no internalized virus, then it would be of the type of uninfected cell. Whereas an infected cell that's beyond the eclipse phase would then be of the virus-releasing cell type. And then for all of the mechanisms that we simulate, you can then see here that these serve as identifiers for the various mechanisms that we model. So in our case, we do allow for superinfection, in which case, whether or not an uninfected cell or an infected cell is exposed to virus, either way, it can internalize virus. Whereas infected and virus-releasing cells can only replicate virus because uninfected cells have no virus inside. Our virus-releasing cell type specifically denotes when an infected cell can begin to release virus into the extracellular space. Both infected and virus-releasing cells release our cytokine field or release into the field, whereas only the virus-releasing cell can die due to effects from viral production. And then killing by oxidative agent from immune cells or the bystander effect. These are indiscriminate with respect to the particular stage of epithelial cell. So both the bystander effect and the oxidative agent can kill uninfected cells and virus-releasing cells or infected cells alike. Now for our immune cell type, we have two types of immune cells, and the two cell types describe the state of a particular immune cell. So for immune cells that have been introduced into simulation, we call these inactive immune cells, and that there are specific immune cell mechanisms that they can't yet do because they've not been activated by cytokine. And with sufficient exposure to cytokine, they then transition to our activated immune cell type. So you can see here in this table, one of the mechanisms we have here is random motility. So we model our epithelial cells as being frozen in that they don't move, whereas the immune cells that sit on top of the sheet do undergo random motility. Whereas only the activated immune cells will actually chemotax towards sites of infection by cytokine signaling. Now, both immune cell types will kill an infected cell on contact, whereas only the activated immune cell type will release cytokines to relay immune cell signaling to the immune system. And then the same is true for the secretion of the oxidative agent. So when an immune cell is in the activated state, due to exposure to cytokines, it can then secrete the reactive oxygen species to kill epithelial cells. For modeling very basic viral replication, the mechanisms that we're particularly interested in for now were getting virus into a cell from the extracellular space, then modeling replication with exponential growth inside of a cell, and then a corresponding release of the replicated virus back into the extracellular space. So this is a diagram of the mechanisms that we represent, these four here being described by a system of ordinary differential equations that's attached to each cell type, whereas we have specific models and events for the cases, or for the events of both virus internalization and then release of internal virus into the extracellular space. To model immune recruitment with our compartmental model, we have our ordinary differential equation, just a single variable that we refer to as S. Now, we say by convention that when S is positive, that there is a net pro-inflammatory state, in which case the probability of immune cells being added into our simulation space increases. And then when our variable S is negative, we say then that the immune system is in an anti-inflammatory state, in which case the immune recruitment model begins to remove immune cells from the spatial simulation domain. Now, with that convention, then we say that when there is an increased amount of cytokine in the simulation space, then there's a corresponding increase in the variable S, making it more pro-inflammatory. And then for regulation for something like a resident immune population, we then say that the number of immune cells that are in a simulation space then decreases the variable S. So then we have four cell death mechanisms that we represent. The first being virally induced apoptosis, and this is due to high amounts of internal virus. The second is contact cytotoxicity, and this occurs when an infected cell comes in contact with an immune cell. The third is the bystander effect, and this is the case when an epithelial cell's neighbor is killed by contact cytotoxicity, in which case it also has a chance of dying. And then the fourth is oxidizing cytotoxicity, and this is due to the secretion of the oxidative agent. In high levels of this field, there's an increasing chance that an epithelial cell dies. So then our simulations begin all with the same scenario, which is all shown here. And this is with an epithelial sheet above which there are initially no immune cells, no virus, cytokine, or oxidative agent. So all three fields are initialized as 0. The immune cell population is initially 0, and we start with all uninfected cells except the center one, which is shown here in green. With the simulation time step and the number of steps that we simulate, then we simulate over 1,000 simulation steps, which corresponds to just under 14 days. And then we selected a baseline set of parameters that, by and large, were an educated guess, some of which are just purely a guess. specifically to produce the sorts of outcomes that we wanted based on sort of intuitive parameter variations. And we specifically looked for variations that would either drive the system towards widespread infection of the virus throughout the tissue, or in the opposite direction towards something like viral clearance or containment of infection. So the baseline parameter set, the way we like to think about it is that it's something like being at the boundary of balance between widespread infection and control of the virus by the immune system, where we started with a parameter set that's just on the side of the boundary where infection propagates. And this is what you can see here on the right. So this is spatial results over almost 14 days. Beginning with our initial simulation scenario and moving in time from left to right, you can see that there's the spread of infection throughout the domain. So again, the uninfected cells are shown here in blue. The infected cells that are not yet releasing virus are shown in green. The red cells are the virus-releasing cells that are actually releasing virus into the extracellular space. And then all the black cells are the dead cells. You can see in this case that this widespread infection moving from left to right eventually leads to complete damage of the tissue due to the cell death mechanisms of virally induced apoptosis, as well as the 3 killing mechanisms by immune cells. This is what it looks like. So up top, this is just the immune, or just the cell populations. And now here is with all 3 diffusive fields. And we also did a reference case where we just completely disabled the immune system. Started with the same initial conditions and observed the way the infection dynamics would work, say with, sorry. And then you notice that in between here, between these green and red regions, then that there's lots of stochasticity. There's lots of variation between these sort of regions of deterministic outcomes, where we can get lots of different outcomes, lots of different events that can occur just over the time that we've simulated. So when we observe these and really delve into the details, we noticed that there are some particular patterns and events that occurred a lot throughout the simulation replicas. And they occurred enough that we gave them names and we defined them. And they're all shown here on the right with these different names. We can go through these very quickly. So there's the case of widespread infection, like here in the bottom left. Like our baseline parameter set, everything is dead by the end of simulation. There's something like slowed infection, where by the end of simulation, the virus is not contained, but it's also not completely widespread, which occurs somewhere in this region. And this is like something like the innate immune response slowing down the progression of infection to where the adaptive immune response has a better chance of controlling the infection. We have the case of containment, where there are no infected cells at the end of simulation, but there's an appreciable amount of extracellular virus in the extracellular space that gives the possibility of new infection sites recommencing infection. There's this interesting event called recurrence that we observed at one of these extrema, in which case we notice that all infected cells at some point in simulation are dead, and then the extracellular virus is internalized elsewhere, and the infection begins again. And in some cases, this occurs multiple times. You can see with these little peaks in infected cells. Then there's a case of clearance, and this is the case where the initial infection is stopped, and the extracellular virus by the end of the simulation is not appreciable. So then from here, we did a second parameter sweep, and this was to simulate something like a drug therapy. So one of the parameters of interest in our ODE description of the viral replication process inside of each cell, this is a parameter that controls the rate of exponential amplification of the virus inside of a cell. So this parameter Rmax, when this increases, this then increases the exponential amplification which is the representation of genomic replication. And so our simulations of drug therapy was then to choose from the baseline parameter value for this replicating rate Rmax, choose some reduction of some amplitude of interest, and then apply it at some moment in time in simulation. So this would be something like a prophylactic treatment occurring at the beginning of simulation if the reduction is applied then, as opposed to a later treatment due to, say, a late onset of symptoms. And so in this parameter sweep, you can notice here, so moving from left to right, all of these dashed lines denote what we call the time delay of application. Now these are the moments in simulation time when a particular reduction in the replicating rate was applied. So moving left to right is earlier to later treatment. Now moving from top to bottom, bottom to top, these coefficients correspond to the reduction in the replication rate that's applied at a particular moment in time. So here at the lowest, 0.875, this is a 12.5% reduction in the original replicating rate, whereas 0 means that all genomic replication at whatever time it was applied, all genomic replication stops. And as you would expect for early treatment and very strong treatment, which is here towards the top left, this is counting the number of uninfected cells. And you can see here we simulated 900 epithelial cells for all simulations, and in most of these these curves are practically unchanged. These for very early treatment and absolute treatment, complete stop to genomic replication, but practically nothing happened, which is what we would expect. Whereas very late and very weak treatment, shown here in the bottom right, see all uninfected cells go to 0. And again, we see the same sort of deterministic behavior for the lots of stochasticity that we simulate. We can find these parameters at these extreme ends of the parameter sweep where there's a certainty of outcomes, and the simulation replicas behave about the same way in time. And then again, in between, we have these sort of fuzzy boundaries between those two regions of deterministic outcomes, where we get lots of variability, even very clear cases like here of a bifurcation, where in some replicas, you see that all of the number of uninfected cells goes to 0. And with the same parameters, instead, we get something like delayed infection for other replicas. Now, one of these parameter sets in particular showed some very interesting things that really demonstrates why this happens and why this is possible with the sort of modeling that we're doing here. So these are 3 different replicas of the same parameter set. The first one being a positive, what we've called a positive outcome, which is that at the end of simulation, there's at least an appreciable amount of uninfected cells, which you can see here. Not many infected virus-releasing cells, whereas in the negative outcome, there's practically widespread infection. Not, and I think in this case, not any uninfected cells by the end of simulation. And then we have something in between. And what we found here is that the difference, when we really looked at the numbers, with the metrics that we took, total extracellular virus, the number of all of the different cell types, we found that the difference between the positive and intermediate outcomes was due to a delay at the initial site of infection, meaning the first initial cell of infection. And what happened here is that the first cell that was infected died, but it died after it had already released virus into the extracellular space, in which case recurrence occurred at a neighboring uninfected cell, and then the infection progressed. Now this matters because these are simulations with the same reduction in replication rate being applied at the same time, and with this delay in the start of the actual initial infection, this then makes this treatment at this time more effective. Now this didn't occur, this delay didn't occur in the intermediate outcome, and we got something like this with delayed infection. Now, the difference that we found between the intermediate and the negative outcomes occurred much later, around, I think, 4,000 minutes. Now, the only difference that we really found when a difference popped up between these two simulation replicas, the number of cell types were all approximately the same, whereas there was a difference between the amount of total extracellular virus. And the only explanation, at least that we can imagine, is the differences in spatial effects, where the cells were infected and the nature of diffusion, even for the same number of infected cells, those infected cells may be in different stages of viral infection, which means that they would then be releasing more or less amounts of virus. And that is quite literally the only way that we know how to explain that difference. And so we say that the differences here are due to the high sensitivity of the system to stochasticity for very early infection, and then the effects of spatial distributions of infection. And so in summary, our takeaways from all of these results is that we can find extrema in this very large parameter space where we do get deterministic outcomes. Simulation replicas, you can simulate it over and over and over, and you can get approximately the same behavior. But then there are regions in between those deterministic regions where it's like a fuzzy boundary, where you can run multiple simulation replicas and you can get different outcomes. You may get two sets of outcomes, but the same parameters and the same initial conditions can produce multiple outcomes. And these are by and large due to stochasticity associated with these cell-based events, the subcellular processes, as well as the spatial distribution of the infection as it progresses or doesn't progress. And then when we simulated drug therapy, we found particularly that early treatment is better, which is not particularly surprising. But we also found that some of the lower doses, when applied early, or still have the ability to be effective. So this is our NanoHub tool, which hopefully many of you have now managed to get registered for and loaded. And this is the baseline parameter set. So this is CompuCell Player. This plays model specification files. And this is the cell field for the epithelial cells. This is the cell field for the immune cells, which again is one level up from the epithelial sheet. And then we have a diffusive virus here. And we can watch this go. This is the This is plotting values of the ordinary differential equation that simulates the immune response. So again, when this is positive, then there's a pro-inflammatory state, which will introduce immune cells to the domain. When it's negative, this will then begin to remove immune cells from the domain. And then as infection progresses, cytokine signaling, particularly by the infected cells, will increase. And that cytokine value will then be mapped onto increases in the state variable, representing the inflammatory state. And you can see that it's starting to increase. So here, there's a minor difference. Blue cells here still show uninfected cells, and green is infected cells. Red are still virus releasing, but in this case, the dead cells aren't shown as black, they're shown as yellow, just to make it a little easier to see. So one of the things you'll notice here is that this is not something like pure diffusion, where there's a complete spherical symmetry, but instead, due to the stochasticity and the modeling at the cellular level, we end up getting these sort of sparse distributions, something more like what you would think of in discrete diffusion. One of the other things you might notice when watching this, if you're really focused on the progression of damage and the distribution of the dead cells, you'll notice that there will be like this sudden appearance of these new regions where lots of cells suddenly die. And this is most certainly to do with the secretion of the oxidative agent doing killing by the reactive oxygen species, whereas see these more isolated incidents of cell death popping up. And this is due to contact cytotoxicity and then a bystander effect. So if an immune cell happens to wander into contact or is introduced in contact with an infected cell, then that infected cell dies. And then when that cell dies, some of its neighbors, all of its neighbors, also have a probability of dying due to our bystander effect representation. And so, yeah, so we don't need to watch all of this. We'll do, we'll have plenty of time watching simulations, but this is about halfway through the baseline parameter set, but using our NanoHub tool. So that's all set up. So when you load this or the local installation, this will be set up and ready to go to begin working with the baseline parameter set. So this is a good time to maybe pause and do some Q&A. certainly love to hear questions about, or comments with what may have been unclear, what we might have been thinking when we were modeling a particular thing. So yeah, feel free to add questions or comments into the chat, and we can go through those. And then we'll take a brief break, let everyone hydrate, and then we'll come back and dive into this. So Nicole raises a great question or concern. So NanoHub, I believe, currently is configured to just show, I believe, the immune cell level of the cell field and one of the other fields. So the way that you can look at different fields or different levels, you can create a new graphics window by selecting this button here called New Graphics Window. And now with that graphics window or any that you've already created, when you select this drop down, this will allow you to select a particular field that may interest you. And then you can select the planes that you're interested in. So if you go in 3D, again, we're doing quasi 2D, so it's probably most intuitive just to stick with looking along the Z direction, and then you can change the coordinate. So if you are stuck just looking at the immune cells, then you can go down one coordinate along Z, and that'll show you. Great. Yes, so there's a question about, concerning personal research. Yes, of course. Yeah, we would certainly love to discuss this. James, Josh, Giuliano, Gilberto, and I, we all come from, we have a pretty diverse set of backgrounds. So yes, of course, we would love to answer questions about personal research as well. In the meantime, while some people may be typing questions, for anyone else who may just be playing with the NanoHub tool, just preemptively while we wait, you notice there's the steering panel here. We have set up a couple of different parameters of interest for you to be able to vary without touching the code at all. So this will update parameters in real time so that, say, when you increase one of these values, say by a factor of 100, you should then see that suddenly there's a very drastic change in behavior. Okay, so Christian has a question. Is it possible to use experimental data to inform your parameter selection to match your model results with known information about them? So Christian, this is one of my favorite potentialities with this modeling. We certainly believe it is, and in some projects we have done it. I think that James could probably elaborate on that quite a bit. In my particular research, I'm interested in taking these sort of higher level measurements. For example, we'll just roll this back a little bit. So these measurements here are the measurements of total extracellular virus and the total cytokine. And something that I'm particularly interested in is taking these global sort of measurements from simulation and then turning something like machine learning on to finding the parameters of the model that fit those simulation results to experimental data. So I think James could tell you, James could best tell you about cases where groups have done this in the past. That's my particular interest for this modeling project as well as others. Yeah, you're very welcome, Christian. Let's see. Nicole, did we parameterize the ACE2 expression based on experimental measurements? We are doing antibody staining experiments right now to try and better characterize ACE2 expression. That is a very exciting prospect. So Josh can answer this best concerning selection of those numbers. And Josh, feel free to jump in at any point.
So we did when we were calculating the probability of internalization, we did use the parameters binding affinities from literature to sort of inform the probability of internalization in our model. But our model is not explicitly modeling receptor trafficking and explicitly modeling binding. So in a sense, we plugged in those at a very general level at a very conceptual level, but it's not explicitly. But yeah, we did use the available experimental data to sort of like inform some of the values of these parameters.
Okay. Arjan asked, is it possible to make 3D models like neuronal cells and around electrical or biochemical models? So yeah, this is a great question. I mean, it's certainly possible to do 3D models, and we have, I think there were a couple of examples very early in the slides of modeling, say, vascular networks and whatnot. Modeling electrical signals is not something that I've seen done, although I think that James would know best. It's a particularly interesting prospect, though, and I personally would be interested to see more done with that. To my knowledge, it's hard to say that it can't be done. How easy it is to do it is of course, an entirely different question. But we can certainly do 3D models, and we're always looking to add new mechanisms into the framework. Not this particular framework, but into what we distribute with CompuCell. Okay, that's another question. Trying to run a simulation that was created by my previous lab mate in the newer version with some adaptations. When I try opening my simulation in the player, it shows an error. This sounds like an issue of versioning. Yeah, so we try very hard to maintain legacy features and to try to not break old simulations or simulation files. But of course, it's certainly possible. And when we went to CompuCell 4, excuse me, um, there were a lot of potential code breaks because we updated from Python 2 to Python 3. So that may be the cause. Of course, this part of my job description is helping work with that stuff. So please feel free to email me or Jim Sluka, and we'd be happy to help with that. Okay, Nicole, follow-up question. Could you provide a quick reference guide somewhere to what the parameters correspond to? Ah, yes. So these parameters that that are available in the sliders, the preprint describes them. And I will include, just to quickly get that, here is the link again to the preprint. So those parameters are described there, but qualitatively, the first K on, increasing K on corresponds to an increase in viral internalization, or an increasing rate. And this was demonstrated in one of the parameter sweeps. So increasing K on is increasing the probability of viral internalization. Beta delay is the immune response delay coefficient. So for a smaller delay, there's a stronger and faster immune response. And then the replicating rate, or replication rate, that is the coefficient that corresponds to exponential amplification in the viral replication model. So when you increase that value, you should see an increase. Sorry, I was trying to find the equations really quickly. When you increase, I think it's labeled replicating rate. Oh, Rmax. Yeah, so when you increase Rmax, what you'll find is that inside each cell, the genomic replication is faster. So that's what these three do. And we'll, once we come back from the break, we'll dive into these, and as well as a couple of other interesting parameters that make this do some really interesting particular things. Okay. Okay, so a little bit more from Sheraton. Sorry if I mispronounced that. So a little bit more on these two fields. Or it's the same field, but these are two coordinates. So we're looking at what would be z equals to 0 in this cross-section. And then in this cross-section is z equals 1. We're looking at where the immune cells are. So the immune cells stay on this coordinate, and the epithelial cells stay on this coordinate, but they certainly interact. So they interact with the killing mechanisms, where immune cells can induce apoptosis through the 3 different mechanisms that were described beforehand. And then there's also interactions with the diffusive species. So there's cytokine secretion by the infected cells, and a little bit by the immune cells as well. And that cytokine induces activation in the immune cells. So not only are there interactions going on due to contact cytotoxicity at their contact interfaces, there's also the interaction indirectly through the secretion and corresponding response of diffusive species. So yeah, these are not two different cell fields. These are two cross sections of the same field. Okay. And let's see. Now to further that, so yeah, they are non-interacting, and yeah, the second half of this question: can many more layers be added? Yeah, that's a concept that we've talked about a little bit on and off for a while about how to be able to do selective sort of coupling between otherwise non-interacting fields. Since they are interacting, you know, it's sort of a, I guess, a different question then. And then also, yes, cells cannot move in the epithelial sheet. So they certainly do stuff with the secretion or the release of virus and so on and so forth. But for the simplest case, we just fixed these epithelial cells, whereas clearly the immune cells translocate quite a lot. Yeah. So one of the immediate improvements that we've done with this work since we've submitted the paper and built this framework is to unfreeze those epithelial cells. And it's particularly relevant for quite a few things concerning modeling like recovery or selective susceptibility and so on and so forth. But yeah, for the very simplest case that we've done here, they are frozen and can't move. Okay, I think I believe that's gotten us to the end of the questions. So yeah, I'd like to take a break here momentarily, let everyone stretch their legs, grab some water or coffee or whatnot. So let's say you can come back in 10 minutes, and then yeah, then we can get CompuCell fired up, help everyone get their last, actually, yeah, let's do 10 minutes. And then, yeah, then we'll dive into the model parameters and start watching this do some interesting stuff. Okay.
This is James Glazier talking. I just wanted to say during the break, since after the break, everyone will be running the simulation and exploring it. If anyone is unable to run either on their desktop or on NanoHub, please use the yes button in the chat function to ask for help. And so we'll do our best in the few minutes between now and the second half to help make sure that you're up and running so that you're able to do all of the exercises and the examples and explorations for the second half of the meeting.
Give it just another minute or two, let everyone get settled back in. See from the chat, might still be having some issues with getting some CompuCell installations going. So if you're having issues with local installation, say installing it on your Mac, for example, the easiest solution may be going to the NanoHub tool. There's a link to the NanoHub tool. There's a very quick registration process that's painless, and then after that, that should take you to this exact same tool that we were just looking at during before the Q&A session. So for anyone who's really struggling with a local installation, the easiest, least path of resistance might be to try NanoHub instead. And then for anyone who may have missed the links, there is the link again for downloading local installations for those of you who prefer to do things locally. And there may be some installation issues, and if so, maybe try the NanoHub route and see if you have better luck that way. We certainly want to see everyone who would like a local copy. We would certainly like to see those get working. And if it's just not feasible to get it going now, then Jim and I in particular would be happy to help after this workshop to get things set up on your local machine so you can hack away from the privacy of your own home. Okay, so that being said, let's go ahead and get back to it. Yeah, so these links have been provided in the chat. The NanoHub tool, again, is configured such that when you launch it, it starts playing the simulation with the baseline parameter set. Whereas the local installations do not. And for the case of the local installations, you can retrieve the simulation files from one of two places, depending on your preference. Some people, like me, swear by GitHub, in which case you can find the files there. The link has been dropped into the chat. And for those who are more GitHub adverse, I've also included a shareable version in Google Drive. A link to those files is also in the chat. Now, when you download the GitHub version, the entire GitHub repository, the folder, the directory that's downloaded, you can follow this file path, and that'll take you to the simulation files. And we'll go through the process in NanoHub as well for actually opening up the simulation files in our editor, which is called Twedit. So again, please just let us know in the chat if you get caught up on anything, and we'll get all those issues sorted out as we progress. So first things first, for again, for those of you who've been using the NanoHub version, when you launch the tool, Player, CompuCell3D Player automatically opens and starts playing. The CompuCell3D Player does the actual execution of simulation files, does real-time rendering based on whatever you specify. So you can go through the activity of creating new graphics windows, doing new renderings of new fields, and observe what happens as the simulation progresses, as well as, for these cases, using steering panel, which is a way of modifying specific parameters that we've set up in real time. So the simulation will update these parameters as you select them. Now for a local installation, you'll start with something like this. So when you launch Player, which can be done just by launching CompuCell, you'll get to CompuCell3D Player. And the way to open the simulation files in CompuCell3D Player is to go to File, Open Simulation File. Now, if you navigate to whatever version that you downloaded, you'll get to a directory called Model, and in Model, you'll find a CC3D file called viral infection vtm.cc3d. Now, this is what you want to open. So we'll do this, and when we click Open, it should show the file name at the top. Now, in the top left, you see some control buttons. Now, the first one says Play, or Run. It's a play button. And the next one says Step. Let's click Step. So what Step does is it steps the simulation. We break up these simulations into a progression of simulation steps. Now selecting Run will just run through the various simulation steps according to what's defined in the simulation specification files, whereas Step will just go one simulation step and then it'll stop and it'll wait for you. This is particularly convenient when setting the steering panel, because you can initialize a simulation just by clicking Step. And then you can set one of these parameters to something different than the baseline parameter set. Say 10 for k-on. We'll multiply the k-on value, the baseline parameter set, by 10, and then when we hit Run in the top left, now you can see that the simulation is going. And it's going with these values. So we can pause here. You'll notice one of the things here is that this is only showing rendering every 10 steps. So let's pause. And let's go to Tools. And then Configuration. Now the first tab that pops up for CC3D preferences is called Output. And this first option, Update Screen every nth MCS. This is the setting to do rendering frequency, to set the rendering frequency. So when you set this to 10, it'll update every 10 steps and set that to 1. and then click OK. Now, when we hit play or run again, you should see that now it's updating every simulation step. These are controlled by a different setting. This is doing rendering of specific data that's been configured in the simulation files, whereas these are native to player itself. So rendering of spatial data, the frequency will be controlled by that setting. Okay. So let's pause there. And now, let's move to Twedit. So Twedit is a program that we also develop and distribute that is specifically designed to facilitate model specification for CC3D based models. So both in the local installation and in NanoHub, you can immediately launch Twedit from Player by this button here. Called Start Twedit++. And when you click that, it launches Twedit. Now, in Twedit, we'll open the same simulation file using a similar procedure, this time in CC3D project. And we'll go to Open CC3D project. Now, if you navigate to the same location where the CC3D file is located, you can just open that, and that'll open the simulation files in Twedit. Now, for the case of NanoHub, the way to do this in NanoHub, start Twedit with the same procedure, clicking the button right here. That will launch Twedit. Now, to find the project files in NanoHub, again, go to CC3D project in the menu bar and do Open CC3D project. Now, this went here automatically by memory, but for anyone else, notice on the left, you should see a directory that corresponds to your username. When you click on that, you should find a directory called CompuCell3D_demos. If we go in there, you should find a directory called coronavirus. We go in there, and that's where viral infection vtm.cc3d is stored. So in NanoHub, open, and now we're at the same point. In NanoHub, and in my case, also locally. So I see from the chat that there are quite a few people having issues getting started. So let's go through that again at maybe a brisker pace, just to get anyone who may have gotten behind up to speed. Because once we get through this, it's pretty much smooth sailing. So from Player, in NanoHub, Player will open up automatically having loaded the simulation files. For local installations, in Player, you do File in the menu bar, and then Open Simulation File. navigate to wherever you saved the model specification, and open viral infection vtm.cc3d. And if it's successful, then you should see the file name at the window title. And again, to launch Twedit from player, there's a button here called Start Twedit. You click that. That will launch Twedit. It's the same in NanoHub. And then opening the simulation files in Twedit is a similar procedure. In the menu bar, find the item CC3D project. Go to Open CC3D project. Again, find the location for local installation. Find the location where you saved model files and find viral infection vtm.cc3d and open it. In NanoHub, after having launched Twedit, CC3D project, open CC3D project. On the left, see a directory with your username. Should make available a directory called CompuCell3D demos. In there, when my internet catches up, there we go. In there, find coronavirus. And in there, viral infection vtm.cc3d. And open it. Okay, so hopefully everyone is there, whether on NanoHub or locally, should have player running with the simulation files opened, and then Twedit with the simulation files loaded. Should see on the left, should see a little item that says viral infection vtm.cc3d in the simulation pane or project pane. The same should be true for a local installation. Now, to open all the files, double-click that item. Viral infection vtm.cc3d on the left-hand side. Double-click that. That should open a bit of an array of different Python files and one XML. This is in my local installation. And then in my NanoHub tool, it's the exact same. Just double-click that, and it's going to open a lot of simulation files. Now, some of these simulation files, for anyone who would actually like to do development in this, may never even really end up popping up in your universe. But some of them are immediately relevant for today. The one in particular that's relevant for today that we'll actually get into a tiny little bit is called Viral Infection VTM Model Inputs, .py. So this is the input script, the Python script, where all of the model parameters are defined. And the reason that they're all in a separate script is because there are a lot of them. And what we really like to do when doing model specification is to focus on the actual implementation of the models in one script, and really focus on the interactions that we're describing, and then deal with all of this stuff elsewhere at a different time. And so all of these parameters, all of those values, those are all defined in viral infection BTM model inputs.py. It's about 291 lines long. And the local installation, it's the same. And this is configured, again, with the baseline parameter set. So any variation that you make in this, you can then observe the behavior, and then you can look back either at the preprint or at previous simulation runs that you've done, and you can look at the differences between what happened for the baseline and then what the corresponding effect was for modeling, or for simulating a variation in a particular mechanism. And so the first one of interest, let's do some parameter variations using the steering panel. So we have already previously talked a tiny bit about this parameter, K-on. It's one that's available in the steering panel. So this parameter corresponds to internalization, such that when you increase it, when you increase the value, the rate of internalization will correspondingly increase for a given amount of extracellular virus that a particular cell is exposed to. So for those of you who don't particularly care for the steering panel and are interested in the code, you can find this at line 264 in viral infection model inputs.py. That's all the way down here. Yeah. But for our intents and purposes, let's increase this, say by a factor of 100. So let's have everyone, if you have a simulation loaded in player, let's hit stop if it's available. If it's not available, then it's not running. So hit stop in player. So this will reset the simulation. And now let's hit step to initialize a new instance of the simulation. Okay, so now that these screens have all popped up, everything is now initialized, but it's paused because we've only done the first step. Now from here, let's go to our steering panel and let's increase this K-on value by a factor of 100. So if you click on the drop-down, go all the way to the bottom item and set it to 100. And that's going to take the baseline parameter and multiply it by 100 as soon as we hit run. Now what we should see is that for a given amount of virus, if the internalization rate increases, then one would expect that the propagation of infection should be faster. Now, we already know from the baseline parameter simulations that we went through before the break that for those parameters, widespread infection already occurs near the end of simulation, which is 1,000 simulation steps. We would expect then that increasing the rate of internalization would then make widespread infection occur much faster. So let's see what happens. Let's hit run. And of course, you can set up new graphics windows to look at different things as you please. We've been particularly interested in the location of the immune cells, the location of the various infected cells, and the distribution of the virus field. But you can create new graphics windows with this button here, and then you can select different results that you would like to look at. Just make sure that if you do change one of these settings, say selecting a different result or looking at a different level, this may pause player, and you may need to just hit run again. And that'll just resume where the simulation was paused. So we see here on my screen, I'm at about 150 simulation steps, and we see a lot of infection. This is happening very rapidly. So we see a lot of infected releasing cells, a lot of virus releasing cells. And now a lot of the infection front spreading much faster. We're at around 250 simulation steps on my screen, and this is almost halfway to complete widespread infection. That is much faster. The baseline parameter set required almost all 1,000 simulation steps. Yeah, you'll also notice that the distribution of infected cells tends to be a bit more sparse. There's an infected cell, say, out here on my screen. This is pretty far away from the actual front of the main front of infection. Now, the reason for that is that that cell is much farther away than the main front of infection. It's more likely farther away from the main infection front. It's more likely for internalization to occur due to this variation in this value, this parameter K on. That means that the probability of infection, of internalization occurring increases everywhere, meaning that new infection sites farther from some main wave has a greater chance of occurring. Now you'll also notice the number of immune cells is a lot. It's a lot more than where it started, which was at about 5. You'll notice that in the immune response model, the state variable S is very high through here. And that's due to cytokine signaling that's, well, most of the cells are now dead, but at one point there were lots of cells that were secreting cytokine, and there's a corresponding effect in the immune response. And that immune response then, when very high and positive, introduces more immune cells. Now, if we pause here, so on my screen, I paused at 642 simulation steps. And all but 1, 2, 3, 4, 5, 6, all but 6 cells are dead by 642 simulation steps. We can see an appreciable change in this, just this qualitative outcome of everything dying. It's not really qualitative, but it's a categorization. It's a categorical result, which is everything died. And for the baseline parameter set, this occurred anywhere from 900 to 1,000 simulation steps. Whereas when we increased the K on value by a factor of 100, you see that it happened much faster, which is about what we would expect. And then for everyone's reference, in these simulations, 1,000 simulation steps is about 14 days. And the exact discretization we use is that every simulation step corresponds to 20 minutes. Okay. Now let's hit stop. I should also mention, I forgot to mention this beforehand for those of you who like doing the actual parameter variations in TWEDIT or in the code. To have done this same parameter variation in TWEDIT at line 264, this is the baseline parameter set. It already has a multiplier of 100 on the end. That's not something that I've added to do the variation. That's part of the calculation to do the baseline parameter. So please don't be confused by that. But to have done that 100 fold increase programmatically, we would have just gone to line 264 and typed times 100. And then hit save, or just whatever your hotkey is, minus Control S. So multiplying that by 100, that does the same variation as what I showed setting up in the steering panel. The only difference is that to do this programmatically, you have to, after doing your modifications, you have to hit save. And then you have to restart the simulation. So making programmatic changes manually don't get updated mid-simulation. Rather, to do a modification in the actual scripting, you have to make the modification, save it, and then reload the simulation. Okay, now we expected that infection would be faster on the tissue scale. The viral infection would occur faster because internalization at the cellular level was faster. Let's see if the opposite is true. So now let's go back to our K-on multiplier at the steering panel. And let's set this instead to, let's do 0.1. I'm sorry, 0.01. So we'll divide this by 100 now. And then let's hit stop in case anything is loaded and running. And we'll reset the simulation. So K-on multiplier of 0.01. And let's hit run and see what happens. Oh, I'm sorry. We need to first do one step. So we will hit stop and then step. Now that everything is loaded, go to the steering panel, go to K-on multiplier, and set the factor to 0.01. And now let's hit run. So we see that virus is being released, and that's due to replication in the initially infected cell. There's the transition over to the virus releasing cell type, at which point secretion or release of the virus occurs. But nothing else is happening. So I'm now on my screen, I'm at I'm past 100 simulation steps now, and the first infected cell just internalized. Meanwhile, my initially infected cell just died. There are no immune cells around there, which means that this first cell died by virally induced apoptosis. So the first cell died, and after having released some virus before it died, so far, now by 200 simulation steps on my screen, I only have one infected cell. And it's only infected, it's not virus releasing yet, which means that the only little bits of virus that were released into the extracellular space are now diffusing and decaying. Now, since the K-on multiplier has been reduced so much, again, this is the parameter that affects the rate of internalization for a given amount of exposure to the extracellular virus. This means that with this reduction in the K-on multiplier, exposure to this extracellular virus is much less effective at inducing internalization. So I'm now coming up on 300 simulation steps on my screen. I still only have that one infected cell. Now, since there's only one infected cell and there's not much in it, there's not much cytokine signaling going on, which means there's not much of an immune response, and the little bits of extracellular virus that were initially released are decaying. So this is just really boring. And as we should probably expect, because internalization isn't happening, and that's a critical step in propagation of the infection. So let's stop that, because that's not going to do much more. Now, let's do something with the immune system. Another critical parameter, one that is very important to modeling immune cell recruitment, is the parameter that affects delay, the timing between when cytokine signaling really starts by infected cells, virus-releasing cells, and when there's a corresponding response by the immune system that then introduces immune cells to fight infection. So this is cast such that when beta delay, which is the second parameter in the steering panel, when beta delay increases, the delay in immune cell recruitment correspondingly increases as well. So for those who like to do this programmatically, this coefficient can be found at line 281. It's called IR delay coef. Again, you can make that modification, save it, and then start a new simulation. Excuse me. For those of us using the steering panel, we can hit stop and then step to start a new simulation. And then you can go to the steering panel, and let's decrease the delay. Let's multiply beta delay, the baseline parameter set, by 0.01. So we'll do another extreme reduction here. So beta delay multiplier, 0.01. And now let's hit play. You're having trouble managing all of your different window panes. While player is running, you can go to window and then tile. And that'll organize things very nicely for you. So we see some infection starting from the initial infection site. There's some release of the virus from the first initially infected cell, which is the only one that's virus releasing. Now what we should see is that as infection propagates, we should see the immune response state variable S. At some point, this should become very sensitive and start introducing a lot of immune cells very quickly. So let's see if that happens. These things are stochastic. Many of these models are stochastic, and so there's always the possibility that something goes unexpectedly, and we can try it again and see what happens. So in my screen, I see what looks like lots of cell death due to contact cytotoxicity and bystander effect. Now here I see regions of dead cells popping up very quickly. This is an indication of oxidative killing. Now you'll notice, at least perhaps some of you might be seeing the same thing, but on my screen, when I observe the state variable for the immune response model, I see some really drastic sort of peaks suddenly. And these correspond with the number of infected or virus releasing cells in the spatial domain. And S, the state variable, responds very quickly to that. Karen has a great question. Where's the color legend? So in Tools and Configuration, you can change cell type colors in the tab Cell Colors. And then you can also control the limits on the color bars of diffusive fields under Field. Now, at this point, at least for where my simulation is, I'm at around 700, 750 simulation steps. And even with this increased or this decreased delay in the immune response delay, even for a very fast immune response, at least for this variation, I still get widespread infection. And if anyone happened to have gotten something different, that would be particularly interesting, and maybe you could mention that in the chat. to see if anyone else got something else, because it's certainly possible. Okay, so in mine, everyone died. So I'm going to pause mine, stop it. I see, yeah, so a question from Karen. Oh, for the different types of cell death. Sorry, I misunderstood your question, Karen. Yeah, as James mentioned in the chat, so we've added tracking of cell death mechanisms, but unfortunately it didn't make it into what went on to NanoHub beforehand. So for next week, that's a piece of data that we'll also be able to track during simulation. We can see the consistency of what mechanisms are really doing the killing. Now, I had some other variations planned for this, but I would actually like to move on to modifying the replication rate. So again, we have in our model this coefficient Rmax is called the replicating rate, and this can be found at line 145 for the coders who like to do this programmatically. The replicating rate is a coefficient that affects the rate of genomic replication, which is the exponential amplification step of the viral replication model. And this is the parameter that we vary in our drug therapy simulations. And this is available in the steering panel as the third parameter among the three, Rmax multiplier. Now, what we would expect is that when increasing Rmax, that the rate of genomic replication should increase. And so the amount of virus in each infected cell should increase much faster. Quick question from Christian. Yes, the previous simulation, the only parameter that was varied was IR delay coefficient. Yes, it was the immune response delay coefficient. For this one, we're going to play with the replication model a little bit. Now, let's first do a tiny bit of parameter setting, or I'm sorry, preference setting, so that we can look at what's actually going on inside of a cell. So in Twedit, you can go up to the very top, and you'll find at line 14, there'll be a variable called plot VRM data freq for frequency. Now this sets the frequency at which all of the state variables for the bioreplication model are plotted for the initially uninfected cell, or the initially infected cell. So when we set this to 1, let's go back, set that value to 1, and then go back to player, and let's stop our simulation if we still have one running. And then let's hit step. Now you should see a new screen has popped up. And at the top, it should just say VRM. And this stands for Viral Replication Model. And this window pane, this is going to plot the state variables of the viral replication model in the initially infected cell. Now, when we run it normally, without any variations in parameters, it should look approximately, well, yeah, it should look like what you see on my screen here. You'll notice that this variable A, which stands for the assembled amount of virus, this value will hit 1 and then will drop momentarily. And you'll notice that that corresponds to the event when viral release begins. And you'll also notice that at around, say, 100 simulation steps, it should find that the variable R at 100 simulation steps should be somewhere around 24 or 25. Again, without any variations in the replication rate. Now, if we stop and restart by just hitting step, we should find that when increasing the replicating rate, Rmax, by, say, a factor of, let's do 0.5. We should find that the replicating rate makes the state variable R lower. We should find something to the effect of delayed infection. And this is because the production of new virus has been decreased. Just as simple as that. There's less virus available to do infection of tissue. So we should see that there's a corresponding decrease in the propagation of infection throughout the tissue. Okay, so on mine, I see that my initially infected cell has died, and I can tell that from plotting of the viral replication model, that all of the state variables have suddenly gone to 0. Everything in that cell is dead, so everything goes to 0. I also see lots and lots of death. And in my particular replica, I, at 300 simulation steps, no longer have any virus releasing cells, but a lot of dead cells and a few infected cells on the perimeter. Now, I would expect that if Rmax hadn't been reduced, then these cells would get to the state of virus releasing faster, and that this would have propagated much faster. But in this case, replication is delayed, which means that it's going to take longer for these infected cells to get to the point where they can release more virus into the extracellular space. The other interesting thing that I notice is that since the rate of replication has decreased, there's then less cytokine production by the infected cells, which means that there's less of a drastic immune response, because cytokine secretion by infected cells in our model is proportional to the state of the virus inside the cell. And if it's been slowed, then the rate of release of cytokine is also slowed, which means that the corresponding immune recruitment signal is lower. So effectively, this looks something like slowing the simulation down with respect to our time steps. It also gives the immune cells much longer to deal with infected cells. This is, if anyone else has seen graphics panes suddenly go black, this is a bug in CompuCell that's been plaguing me for months. So if you do see that, you've not done anything wrong. You can just create a new window and reopen what you had. And so, yeah, in my case, of course, please enter into the chat if you got something different than me, because that's very interesting to have two processors run the same model specification and get two different outcomes. But in my case, I got at around 750 simulation steps. I have some scattered infected cells, lots of death in the middle, and not a very dramatic immune response. And so what this really looks like is an infection that has its severity has been drastically decreased. This is something that would look like a much less threatening sort of infection. This is something that's more like something that can be dealt with when convenient, rather than something that would be very imposing and something like a life-threatening infection. So much more docile, much less aggressive infection. So now, Giuliano and Josh did a wonderful job of setting up these sliders so that we could easily do this. And then I followed that by wanting to do a parameter variation that I didn't tell them about. So let's do that. So I would like to now do the same sort of variation, but in the opposite direction. We've simulated and we've showed parameter sweeps about a decrease in the replication rate, this coefficient. Something else that's interesting, though, is to consider something like a mutation in a virus, where suddenly, with respect to our, say, a viral strain that our baseline parameter set was modeling, say some mutation occurs that doubles that coefficient, Rmax. Let's look at the difference. Now this will require a change programmatically. Since this is not available in the sliders, we'll have to go into the model input script itself and modify it, but I promise it's a relatively painless process. So in viral infection model inputs, you can go to Twedit and go to viral infection VTM model inputs.py. It should be along the top of your bar in Twedit. Now we will go to line 145. Should find the line numbers along the side of your text editor. You should find the variable replicating rate. So this is the replicating rate, our max. And currently it's at the baseline parameter set. Let's double it. Let's say this is multiplied by 2. And now let's save it. And now let's flip back to player. We still have a simulation running. You can stop it by hitting the stop button. Now this time, we don't need to do anything with the steering panel because we've made our modification programmatically. So in this case, once we've stopped the simulation, we can just hit run. And this will reload everything. And now it's going to run the simulation from start, but with this doubling of our replicating rate. So let's watch what happens. I'm going to tile just to clean up my workspace a little bit. Again, that's under Window and Tile. I'm particularly interested in the infection itself and the epithelial sheet. So I'm going to watch this screen for the epithelial sheet. Now, what we would expect if genomic replication has increased significantly, what we should expect then, one would think, is that this infection should rip through the tissue. It should happen very fast. There should also be a more drastic response by the immune system because the virus is being produced faster. Cytokine production is proportional to the amount of virus, which means that there's more cytokine production, so there's a stronger signal. There should be a stronger immune response, or at least a faster one. So let's see if that happens. See infection spreading on my screen by under 200 simulation steps. See infection spreading very rapidly. I already see on my screen some cytotoxic killing. I lost my cell field. Regenerate that, okay? We see a lot of virus releasing cells, and in my case then there was a large group of virus releasing cells that suddenly got wiped out by the oxidative agent. So this is like immune cells bombing, like peppering entire regions and killing anything. In this case, they made a really good shot because they killed all of my virus releasing cells. However, in a previous simulation, at least in my case, I had a large group of, like a large area of cell damage and very few infected cells around it, and then not much happened. That was with using the baseline parameter set for replicating rate. In this case, I had a similar configuration, a lot of cell damage, a lot of cell death, and a few infected cells. Except in this case now, the propagation of infection, it's like nothing even inhibited it. It's begun again. I have virus releasing cells again. I have scattered infected cells throughout. And in this case, infection continues, even though there was lots of cell killing by immune cells initially. And that's interesting. This shows, or at least implies, how much of an effect the production of virus, just the sheer numbers of it, and being able to spread like that for the same immune response, even in cases when there are few infected cells, that there's still a much higher chance of infection restarting after an initial effective immune response, which was what I saw in my simulation. Okay, so my screen, I have around 600 simulation steps, and I think it's pretty clear at this point widespread infection is going to occur in this case, which we would expect because the baseline parameter set it produces widespread infection, everything gets infected. And then we increased the replicating rate, so one would expect the widespread infection occurs. In this case, though, it also seems to occur much faster, which we would expect. Increasing the rate of genomic replication should result in faster infection, and in our case, faster widespread infection, complete cell death, complete infection of every cell, and inevitably cell death. Okay. So I got my widespread infection event. I have no more uninfected cells. And I measured that at around 820 simulation steps. So measurably faster in simulation. Okay, so we have about 20 minutes left in our time for today. And what I would really like to do is to close this out with a little bit of time available to go back through and give all of you the opportunity for more questions, again, either about the model or the implementation or how this might relate to your research or research that we could potentially collaborate on. because we very much are looking to do a lot based on this platform. We're very interested in finding new collaborators in quite a few different ways. I mean, we specifically designed this framework to be modular and to be extensible. So as we'll see next week in our second part, and for those of you who like to dig into the code, hopefully, as you'll notice, all of these various modules, the various mechanisms that are represented in the code, they are implemented to be as modular as we could possibly stomach, specifically with the intent to be able to extract those mechanisms, those models, and replace them with better versions or with new ideas. So we have a generic cytokine field where someone to be interested in, say, 10 different cytokines, then replacing that one cytokine field with those 10 different cytokines of interest and substituting how they interact in the current implementation, making those substitutions should be relatively painless. So we've also developed some workflows to facilitate taking ODE-based models like our immune response model or our replication model. We've come up with a lot of workflows and integrated libraries to be able to very painlessly hybridize and take and integrate ODE-based models into our framework for spatial modeling and for multiscale modeling. And we're very happy to work with, as well as support, model development for new modules and for extensions, whether introducing new abstract objects representing various biological components, or just making improvements to the basic ideas that we've laid out. So as we discussed before the break, we're certainly interested in working with experimental data and with experimentalists to generate data specifically for validation, model parameter determination, as well as using simulation results to inform new experiments to do. So we're very, very keen on getting with experimentalists as well. We in Bloomington tend to stick with the computers and like to make friends with those in the wet labs. So we're also very keen on doing cross-platform validation. So if you dig a little bit into, say, our preprint, you'll notice that there are authors on that who work on 3 different simulation platforms. So us in Bloomington, we work on CompuCell, but then we also have friends who develop Morpheus and Chase. We're very keen on doing cross-platform implementation and validation. We'd like to make sure that our implementations are right, and we're also very interested to see how our model in different, say, cell dynamics methods, what differences show up based on things like differences in methods. And lastly, as we've looked to pursue with this event, we're also very keen to use this framework with the modeling aspects, as well as the architecture, developing the actual software and the simulation files. We're very interested in looking to use this as opportunities in education. So we would love to talk about these sorts of aspects now, after the workshop, throughout normal business hours, via email and whatnot. We're certainly looking to connect and to make new projects and new opportunities with what we've gone through today. Speaking of education, so we do a summer course on model development and specification in CompuCell3D. We do a workshop every summer. So this is an intense, I should say intensive, not to scare anyone off. This is an intensive summer school and hackathon that really starts at how to use Python in general, Python programming language, all the way to building models. So for anyone who might be interested in making new CompuCell-based models de novo of your own, the workshop is a great way to get into that. We in Bloomington, James and Giuliano and we contribute with that, and registration is free and so on and so forth. This year, because of the pandemic, we have to do this virtually, so it'll be something like this, but with pre-recordings and remote hackathons. So lastly, yeah, we're certainly grateful for the funding that we received to do work like this, specifically from NIH and NSF. We've also have running very long list that keeps growing of collaborators and partners we've made in lots of different fields, from brute computer science all the way to molecular biology and physicians. So we're certainly grateful for all of the input that they've had and how they've inspired a lot of this work and refined it. So with that all being said, this is the information here on your screen for part 2. So we'll be doing another version of this presentation tomorrow morning, and then this same time next week we'll do part 2. In part 2, we'll actually dive into the code. So we will actually go through in the XML and Python model specification of the framework. We'll actually dive into that, and we'll go through some of the basics of how to generate some of those graphics rendering panes for tracking data, all the way to doing actual basic model development. So we'll go through the exercise of adding cell types, adding diffusive fields, and then taking some very basic mechanisms and taking a simple representation of that and actually implementing it in the code, and then observe what happens. So again, that's the same time next week for those of you who may have joined late and missed maybe some of the earlier parts of today's presentation. We'll be doing this again tomorrow at 9 a.m. Eastern, and you're welcome to join then as well. So with that being said, I would like to turn this over to all of you, and then of course James, Josh, Giuliano, Gilberto. We would love to take any questions about the model, the implementation, the simulations, any of that, and hear from you. So please, anything in the questions, anything in the chat. And for those of you who are exiting, thank you very much for being in attendance and yeah, for joining us today. It was great to have you. We're very, very grateful for your interest and your attention. Christian has a question. Is it possible for users to define their own fields? For instance, if I wanted to implement a mechanical force field to study, okay. Yeah, so so CompuCell provides a couple of different ways to go about doing modeling with fields. I mean, for biological modeling, spatial modeling, the most obvious mechanism to include in sort of like standard support is reaction diffusion. And we have a whole suite of PDE solvers that do those sorts of things. But we also provide a more generic way of setting up fields with either scalar values or vector valued fields, and then mapping whatever you like that goes on in your field that you've created, and then mapping that onto where the cells go or how they're compressed. The underlying cell dynamics is the cellular POTS model, the GGH model in CompuCell. And that uses a sort of Hamiltonian approach to do things like change where cells are, what spaces they occupy, and so on and so forth. And so the long and short of it is that CompuCell includes a generic way of including additional terms that affect the Hamiltonian-like mechanics of the cellular POTS model based on whatever information you have in some field that you've created. So we have a collaborator in St. Louis who has been very interested in doing something like actual mechanics and doing force calculations and mapping that onto the actual cellular dynamics of the POTS model. And so the long and short of it is, yes, you can create fields. Yes, we provide generic ways to map that onto, say, directed motility or compression or whatnot. And some of us, like me, are particularly interested in improving those models. And yes, James is included in the chat. Yes, this is a very good reference. Thank you, James. And thank you, Christian, for the question. James or Josh, Giuliano, Gilberto, are there any say closing remarks, or I'm sure I missed something, perhaps lots of things that you would have liked to have heard or that I was supposed to have said that I forgot. Of course, is there anything that you would like to see discussed or mentioned?
I just wanted to say, if anybody had trouble getting running and therefore missed part of the talk, again, it's being repeated tomorrow morning, and so you're welcome to join in, especially if you couldn't do the examples and work through them because you were having trouble running. If you join at 10:15 tomorrow morning, you can go through the second half of the talk again and repeat those exercises if you like. We're also all available to discuss collaborations, opportunities, either in virus modeling or elsewhere. Our emails are all available on the website. I also sent them in the chat. And we want to thank all of the people who've helped us with this work over the years. Many of them were on the call today. Maciek Swat was here, who was the original author of CompuCell3D, and he was helping out. And Gilberto Tomas from Brazil is here helping out. So we do appreciate that. We hope we'll see you either tomorrow again or next week. And we're always available to talk one-on-one with people about these or any other projects. Of course, the work that we presented here was done in collaboration with a large number of people. TJ listed quite a few, but that doesn't exhaust the list. There are many more that we would like to thank.
Okay, well, got a couple more minutes, but I don't see too much movement in the chat. So in that case, let's say we could probably go ahead and close this out. The parting and the very last thing I would like to say is that you can follow the project on GitHub. So everything that we do with this project, we update this very meticulously. So you can stay up to speed, as well as fork this repository and do your own developments, which we of course invite and would also love to assist with and support however we can. And then to reach us, to find CompuCell, again, you can just visit CompuCell.org. Our contact information is there. And James has just included both his email and mine as well to discuss anything else after this event. So, otherwise, thank you all very much for your time and for your attention. And we hope to see you either tomorrow or next week.